contraction, let's try to understand the structure of the skeletal muscle. You know that the primary and the fundamental function of skeletal muscle is to provide what is called as mechanical support. There are three fundamental types of muscle: skeletal, cardiac and smooth muscle. Two are known as striated because when you put this muscle under the microscope you see striations. Those are skeletal as well as cardiac. Smooth muscle, when you look at the microscope you don't see any striations. That's why it is called as non-striated muscle. Muscles are like transducers. Name one example for a transducer which converts one form of energy into another form of energy. What are the transducers you know which we use in day-to-day life? Car, which converts chemical energy into mechanical energy. So all the motors, all those things, the switch, light, all those things, electricity is converted. So all these are transducers. When it comes to muscle, they use chemical and electrical commands and they convert into mechanical so that you will be able to move because of this. So that you will be able to support yourself against the gravity. If this doesn't happen, the locomotion would have been not possible. Why? Because you must have seen people who are paralyzed, right? So whatever the cause may be, they in battlefield they must have got a gunshot in their spinal cord, and through which the commands and the traffic or the signal from brain which is conveyed to various parts of the body of the muscle. So if this traffic is interrupted, if this route is interrupted, muscle don't get command to work, they undergo paralysis because all these skeletal muscles is under your will and wish to contract. Now it is no more receiving the command from the higher center, then he or she who is paralyzed will not be able to contract the muscle on their wish and will. So they are paralyzed. So that's why it becomes so important to understand how this muscle undergo contractions. And is there any subtle differences when you come to cardiac, skeletal, and smooth muscle? That we will look at it later. Now, each muscle type dictates inherent differences in terms of rate of contraction, duration of contraction, as well as its metabolism and fatigability. What do you mean by fatigability? Tired, getting tired. So which muscle gets tired? Skeletal muscle or cardiac muscle? Skeletal muscle. You know that. You move, walk, run, all those things. Then what happens is, at the end of it, you feel tired. When you come to fatigability, there are two phenomena. One is the central fatigability. Second thing is the peripheral fatigability. Muscle undergo fatigue. What is central? Sometimes you don't do anything. You are not unable, or you are not able to get up from the bed. You see, I'm very tired. Did you walk? No. Did you? All these times you are sleeping. How can you be tired? That is the central fatigability. That phenomenon is a little different. What you are talking about is the peripheral fatigability in the muscle. Okay. Look at this. What we'll see is about 40% of our body is made up of skeletal muscle. Skeletal muscles are made up of individual muscle fibers. They are called as building blocks. Each muscle fiber is a single cell which is multinucleated, because if you look at the embryonic development of this skeletal muscle, many embryonic stem cells which are helping to form these muscle fibers. Where were you, girls? What happened? Coffee? He did not serve coffee? What? Which lab now? Half an hour break period. What lab is that?
We thought we had lab.
Oh, you thought you had lab.
Sorry.
Ah, nice. What do you use the smartphone for? You can do many things in smartphone. You know that? You can also put reminders. Do you know that? Do you know about it or not?
We apologize.
Single cell, it is multinucleated. When you look at the muscle fiber development, because these stellate or the stem cells, they fuse together during development. So that's why in a single muscle fiber you see different nucleus indicating it is during development process many muscle cells must have fused together. So that's an indication. Okay, whatever it is. Now, so each muscle fiber is made up of myofibrils. Each muscle fiber is made up of myofibrils. Each muscle myofibrils are made up of your contractile proteins. So let's come to that. Now this is a biceps. What you see is you take a small piece of bicep and this is called the fasciculus, and many fasciculus made a muscle bundle. Many fasciculus together, put together, it makes the muscle bundle. And if you tease out the fascicle, what you have is the muscle fiber, and these are long single cell multinucleated muscle fiber. Each muscle fiber length of each muscle fiber may vary between some centimeters to few centimeters. Now if you tease out each muscle fiber, what you see is the muscle myofibrils. When you put this muscle under the microscope, you see a clear-cut striations, and this is why it is known as striated muscle. Why do you see these kind of striations? Because in the muscle, remember, there are proteins which are involved in contraction. These proteins are arranged in a definite fashion. That is why it gives a striated appearance. If something is arranged in a definite pattern, so you see that you guys are sitting very nicely, very organized way. So that's why we see a pattern here. If you randomly sit everywhere, if you take out this, you randomly sit, you may not be able to find any pattern. So the pattern what you see here, mainly because what is the contractile proteins or the proteins which are present in the muscles, they are arranged in a definite pattern. Okay, let's try to understand now. Now each myofibril, when you look at this one, it is made up of what is called as a sarcomere. Sarco means muscle, okay? Sarco means muscle. Sarcomere is a segment. So, sarcomere, many sarcomere will made up a muscle fibril. Many muscle fiber will make up what? Muscle fiber. Many muscle fiber will make up fascicles. Many fascicles will make up muscle bundle. So you got the organization now. So when you come to muscle, if we ask what is the basic structure and the functional unit of a muscle, you must say it is the sarcomere. When you go to kidney, what is the functional and structural basic unit of kidney? Nephrons. When you go to brain, what is the basic structural and functional unit of brain? Neurons. So likewise, the skeletal muscle basic fundamental structure, the unit, functional structure and unit of a muscle is sarcomere. Let's try to understand each sarcomere. It has light bands, it has some dark bands, and what are these due to? I told you it is due to definite arrangement of the muscle contractile proteins. Now, before we go into understand this definite pattern, what you see here, let's understand when it comes to muscle, you broadly classify muscle proteins into contractile protein and the regulatory proteins. There are plenty of proteins. We are only interested in 4 proteins. Okay. Muscle has under contractile proteins and regulatory proteins. Contractile proteins: 1. Actin, 2. Myosin. Regulatory protein, you have 1. Troponin, and 2. Tropomyosin. So troponin, tropomyosins are the regulatory protein, and actin and the myosins are your contractile proteins. These proteins are arranged in a definite pattern. That is what it gives a sarcomere, light and the dark band. That is what it appears under the microscope as striations. How to define a sarcomere? Sarcomere is an area which falls between 2 lines, and these 2 lines are indicated as Z lines. Now this is Z line or a Z line. So if you're an American, you say Z line. If you are UK, you say Z line, and Indian always say Z line because we were influenced by Britishers anyway. So Z line is a line which is made up of a particular protein. It is called as alpha actinin protein. And this line is made up of this alpha actinin protein. Alpha actinin protein acts as an anchoring protein for other other proteins. Now look at this one, alpha actinin protein. Here there is a Z line and here there is a Z line. To alpha actinin protein, filamentous actin, this is the actin, which is a contractile proteins, are anchored. Filamentous proteins are anchored. On either side to the Z line, filamentous proteins such as actins are anchored to this Z line. Z line itself is made up of a protein called as alpha actinin. Remember, actinin and actin, you should have that differentiation. Okay, alpha actinin is what it makes the Z line, to which what is attached is the actin protein. Now centrally, in between the actin, myosin is a larger protein. It is attached. It is present. Myosins are held by a central M line. That means to say, myosins are held or myosins are present or surrounded by, if you look at the three-dimensional picture, myosins are centrally placed around which the actins are there. Actins are attached to Z line. Okay. Now do you see, this is a complete one sarcomere which has actins on either side attached to one Z line here, another Z line on the right side. Centrally, myosins are placed where you have overlapping of myosin and the actin. When you pass a light through that section, because myosin and actins are overlapping, light does not pass through easily. It appears as dark band. Agreed? If these two are not there, myosin and actin are not there, only myosin is there, or only myosin is there, or only actin is there, then when you pass through that, when you pass through a light through that, so what happens? It appears light. So that is why it is called as light band. So you have interdigitation of dark bands, and the light band is the one which gives striated appearance, because these muscles are arranged in a definite pattern. Okay, what about troponin tropomyosin? Troponin tropomyosin, although we have shown here, I am not going to comment at this point of time. So when we look at the molecular regulation, we will look into how this troponin and the tropomyosins are arranged. So you got this dark band, or what does it do to, what does this light band do? So light band, it is also called as isotropic band. Dark band is also called as an isotropic band, A band or I band. So if you want to mathematically represent a sarcomere from Z line, this line to this one Z line, it is called as one sarcomere. So how many different lines it may have? It may have a half of I band, another half is here, and plus one full A band, Now, myosin is fixed. Remember always, it is the Z line to which actin is attached that can move close or it can move further. Suppose if the Z band moves closer, then what happens to I band? If the Z band, now remember here, if the Z band here, Z line, okay, this is Z line, it moves closer. and this Z line moves closer. So what may happen to I band? I band may be obliterated or disappears. Agreed? Okay. So if Z lines are extended further, Z lines are extended further, so what may happen to I band? I band may disappear or it may reduce in its length. So what happens to I band? I band may, size of the I band may increase. So that is what it indicates when the muscle undergo contraction. So this I and the A band, length of this I and A band can vary. Pick her up. It's a nice story too, this thing, right? Okay. Now look at this one. You can ask me how these myosins, are you okay with this? Are you okay till now? Are you okay?
Yes.
I should ask them, are you okay?
She is.
Okay. No, I'm asking her. I'll come to you when I feel like it. We are very specific. So look at this myosin bands. The myosin bands are held in place. Actins are held in place by alpha actinin, that is Z line. And look at the myosin, which is floating, right? Something has to hold this myosin in place. Look at this, another protein. There are plenty of proteins which are, but I'm only talking about very important for your purpose. Okay? There are these titin filaments which will hold these myosin to the Z line tightly. Apart from that, so the actin, now look at the contractile proteins. Myosin is a heavy protein which is the molecular weight somewhere around 460. Actins are light proteins. The molecular weight is 10 times lesser than the myosin. You have tropomyosin, which is about 70 kilodalton, and you have troponin, which is slightly, which are very small proteins. The actin and myosin side-by-side arrangement maintains large amount of filamentous protein called titin. Now look at this titin. This is what we are talking about. The titins is the ones which keeps myosin in place. A larger protein called dystrophin forms a rod that connects the thin actin filaments to the plasma membrane of the. Now look at this one. This is an actin protein. Okay. Myosin is held by one protein called titin, just to keep the myosin in place. And actin also, it is held by Z line, agreed, it is made up of alpha actinin, but in other places there are some proteins. What will happen is, can I write this one?